Hey friends, our today's topic of discussion is transformation in plant cells. Transformation in plant cells. Okay, in plant cells, the transformation can be by two methods. It can be vector mediated or it can be by vectorless method. So first we will discuss about the first method, that is vector mediated transformation in plant cells. So guys, what is transformation first? When we transfer any foreign DNA into the plant cells, it is called transformation. Okay, whenever any exogenous DNA is introduced into the plant cells, it is called transformation. Okay, and one of the method of transformation is vector mediated transformation. In plants, the method for vector mediated transformation is via agrobacterium. So we say agrobacterium mediated transformation. So guys, first quickly I'll tell you about what agrobacterium is. They are natural genetic engineers and they have the DNA called tDNA. Okay, they have a plasmid known as Ti plasmid, which you can see here in the picture. Into this plasmid there is a region known as tDNA. Okay, and this tDNA have and this Ti plasmid have several regions. What are these? These are virulence genes. These are origin of replication, opine catabolism. Please remember this opine catabolism gene is within the Ti plasmid. It is not within the tDNA, right? So opine catabolism, virulence genes, and the tDNA region. This tDNA region have the left border, the right border, and the opine synthesis gene and the oncogenes, which is auxin and cytokinin. These oncogenes are responsible for tumor formation. Okay, this Agrobacterium tumefaciens is the bacteria which forms tumor in the plant because they are able to synthesize auxin and cytokinin in large amount, which causes the unregulated division. So what we understood, that this Ti plasmid which is there in the Agrobacterium tumefaciens, it has a ori region, virulence gene vir region, it has opine catabolism gene and the tDNA region. The tDNA region has the left border and right border. Oncogene, which is auxin and cytokinin responsible for formation genes, and the opine synthesis gene. Got it? Next, we will discuss about the genes which are required for the transfer of tDNA. Correct? Let's begin. So first of all, the transfer and integration of this tDNA into the genome of the plant cell. You can see here that this one, this pink colored box is the agrobacterium cell, which has the Ti plasmid. It has the Ti plasmid contains the tDNA, and this green colored part is the plant cell. So first of all, the transfer and integration of this tDNA into the genome of the plant cell, it is mediated by several genes known as vir genes. Correct? So these vir genes, which are responsible for virulence, they are organized into several operons, which is vir A, vir B, C, D, E, and so on. We will discuss one by one. And these vir are located where? They are located on the Ti plasmid. Understood? And next we need to discuss that which of the genes are required for what purpose. So first of all, vir A and vir G, which is written here, I'll just mark it. So first of all, vir A and vir G genes, these are the genes which will be expressed constitutively all the time. These genes will be expressed constitutively, and the other vir genes, okay, like B, C, D, E and F and all of these genes, they are expressed when the signal is received for the transformation, right? So first of all, this vir-A, let's start with the first gene that is vir-A. This vir-A is a kind of sensor kinase. This vir-A is a kind of sensor kinase, and this vir-A, it spans the membrane. This vir-A is a sensor kinase. It spans the membrane. I'm writing few keywords in short. It spans the bacterial membrane, agrobacterial membrane, and this vir-A, they are the receptors. Receptors for what? Receptors for these molecules. What are these molecules? They are phenolic compounds. What is the name of this phenolic compound? It is acetylsyringone. So first of all, vir-A, which is a sensor kinase, which is a receptor for the phenolic compound, that is acetylsyringone, and where from these acetylsyringone are released? They are released from the plant. Okay, they are released from plant. You can see that these molecules are released from the plant. Understood? Which plants? From the wounded plants. Coming to the next gene, that is vir-G gene. That now this vir-A and vir-G, they are the two component signaling system, right? This vir-A and vir-G, this vir-A and vir-G, they are two component signaling system. So where did you study about two component signaling system? Anywhere else? Please recall. Those whoever can recall and those whoever cannot, please read about it, two component signaling system, and write the answer in the comment box. Okay, so these are the two component signaling system, vir-A and vir-G gene, and they respond to different signals. What happens whenever acetylsyringone binds to vir-A? Okay, whenever acetylsyringone binds to vir-A, this vir-A becomes autophosphorylated. So vir-A is a receptor. Whenever acetylsyringone molecules binds to vir-A, this becomes phosphorylated. Okay, and where is this phosphorylated? It is autophosphorylated on the histidine residue. On the histidine residue, it is autophosphorylated. I'm writing here in short. Autophosphorylation of vir-A. Got it? Number one. First, in the first case. Now, autophosphorylation is on the histidine residue. Now, this phosphate group from the vir-A, it is transferred to where? Since it's a two component signaling pathway, the phosphate group will be transferred, right? Just a moment. With A. Phosphate group is transferred to where? To vir-G. Understood? And this vir-G gene, okay, this vir-G gene has the aspartate residue. So first of all, here this histidine residue which was phosphorylated in case of vir-A, and in case of vir-G, the aspartate residue will be phosphorylated. Okay, the aspartate residue will be phosphorylated. This is the sensor kinase, and this is the response regulator. As you know that in a two component signaling pathway, there is one which is sensor kinase and another which is response regulator. Okay, now this vir-G gene, when it is phosphorylated, this vir-G gene is activated, and the activated vir-G gene, it acts as a transcription factor. Okay, now vir-G gene is activated, G gene is activated, and this activated vir-G gene, what it is going to do, it is going to, first of all, it's protein product, it's a transcription factor, and this transcription factor is the activator of other vir genes. So first of all, what are the initial few steps? The first step is that vir A and vir G gene are constitutively expressed, and vir A is a sensor kinase. It spans the agrobacterial membrane, and it is a receptor which senses the phenolic compounds released by the wounded plants, and these phenolic compounds are what? Acetosyringone. Okay, once vir A is activated, it activates the vir G gene, and vir A and vir G, they shows the two component signaling system. So first of all, whenever acetosyringone binds to vir A, it becomes autophosphorylated on the histidine residue, and this phosphate group is transferred to the response regulator, which is vir G gene, and this phosphate group is transferred to the aspartate residue of vir G gene. Once this vir G gene is phosphorylated on the aspartate residue, they are activated, and once they are activated, this vir G genes are the transcription factors which are the activator of other vir genes. Understood? Next, we move forward. After that, what will happen once vir G gene is activated, right? Once vir G gene is activated, the second step is that this vir gene is the transcription factor which goes and binds to the promoter. Okay, it goes and binds to the promoter of other vir operons. Okay, this vir G gene, what it's going to do, it will activate the other vir genes. How? By binding to promoter of other vir operons. By binding to promoter of other vir operon. Understood? In that case, other vir genes will be activated. So the first vir gene which is activated after vir G gene or vir G transcription factor binds, first of all, what will happen? The next part is that this vir D1 and D2 will be activated. Vir D1 and D2 genes will be activated. As you can see in this picture, that next is the don't go with the steps written in the picture, follow what I am saying, that where D1 and D2 genes, they are activated. And what is the role of this where D1 and D2? They will generate single stranded tDNA, because single strand is going to enter into the plant cell, not the double strand. Okay, they will generate single stranded tDNA. I erase and write properly. They will generate single stranded tDNA. How are they going to generate? Let's see. This vir D2 acts as an endonuclease. What it's going to do, it is going to cut the phosphodiester bond. Okay, it's going to, since this, if this is a Ti plasmid, this is a double stranded Ti plasmid, this D2 is going to cut the phosphodiester bond. Okay, if this is a T plasmid and this is a Ti plasmid, we say double stranded Ti plasmid, this vir D2 is going to cut the phosphodiester bond. Okay, and after it is cut, it remains attached to the nicked DNA. Okay, they remain covalently attached to the nicked tDNA. Nicked, correct? And they are attached to this tDNA via tyrosine residue. Attached to nicked tDNA via tyrosine residue. Understood? So this is the tDNA. Just a moment, guys. So this will D1 and D2 They are responsible for generating the single stranded tDNA. This TD2, just a moment. So this vir D2, they remain bound to the 5 prime end of the tDNA, right? They remain bound to the 5 prime end of the tDNA. This is D2. And they remain bind to the 5 prime end of the tDNA. And guys, what's the role of this D2 while it is bounded to this 5 prime end of the tDNA? See here, what is the role of the D2? So first of all, when vir D2 remains bound to the 5 prime end of the tDNA, it indicates that it might prevent the tDNA from exonucleolytic attack. So it prevent the tDNA, one strand it is going to prevent because they are going to make single stranded tDNA. So it is preventing the tDNA strand from exonucleolytic attack. From exonucleolytic attack. Okay, to the 5 prime end, and also to distinguish that this is the leading end. Also to distinguish that this 5 prime end is the leading end. What is the meaning of this? Because this is the region from where the tDNA will take entry into the plant cell. Okay, so this is the leading strand. So it also indicates that this 5 prime end where the with D2 is bound, it is the leading end during the tDNA transfer. It is the leading end of tDNA during transfer. Got it? So I again recall the role of with D1 and D2. With D1 and D2, what they are responsible for generating the single stranded tDNA, and this single stranded tDNA is said to be as T strand. Sorry for the writing, right? And this tDNA, sorry, and this with D1 and D2, what they are going to do, they'll generate the single stranded tDNA known as T strand, and D2, especially the D2 acts as an endonuclease. What is going to do? It will cut the phosphodiester bond. It will cut the phosphodiester bond, okay? And they remain covalently attached to the 5 prime end of the tDNA strand via the tyrosine residue, okay, so known as phosphotyrosine bond. And this D2 is seen to be preventing the tDNA strand from exonucleolytic attack, and also it indicates that this 5 prime end is the leading end of the tDNA during the transfer. Okay, next we come to another virgins. So next comes the role of vir C. So we discussed vir AG, we discussed vir D1, D2. Next comes the role of vir C. Vir C1 and C2. Okay, what are their role? This vir C1 and C2, they bind to the overdrive sequences. Okay, so if this is the tDNA, okay, this is the tDNA, one strand will go into the plant cell and the other strand will be degraded. So one strand which will remain intact, which will remain intact, that will remain bind to D2. This is D1 on both sides, this is D2 on both sides, D2 on both sides, This is the left border and right border. This is the left border. This is the right border. Okay, this is the right border. This is the left border. Right. Now, towards the right border, there is a region. Towards the right border, there is a region named as overdrive sequences. Here, there is a region named overdrive sequences. And to these overdrive sequences, Vir C1 and C2 is going to bind. Okay. To this overdrive sequences, which Vir genes bind? It is the Vir C1 and C2. So Vir C1 and C2, they binds to right border. I'm changing the color of Vir C1 and C2. This is the one. Okay, this Vir C1 and C2, which are these, they bind to the overdrive sequences. Where are these overdrive sequences? They are present near the right border. Present near right border. Okay, they are present near the right border. And what is the role of these sequences? So this near the right border of this tDNA region, this is the tDNA region. Near the right border of this tDNA region, there is the cis element. There is the cis element. This overdrive sequences is what? It's a cis element. Okay, and this is required. Binding of C1, C2 is required or on the overdrive sequence because it helps in what? It helps in efficient tDNA strands transfer. Efficient T strand transfer. Understood? Where are they located? What is the role of C1 and C2? Right? Next we come to another Vir gene. So this tDNA segment has been formed. Now this tDNA needs to be transferred to the host cell, that is the plant cell. How it will be transferred? It will be transferred to the host cell via a secretion system. Okay, via a membrane spanning complex, right? So whenever the T strand comes out from the agrobacterium, it has to cross several membranes. What are the membranes? There is the plasma membrane. Okay, these T strand has to cross several layers like plasma membrane, like peptidoglycan wall, like the outer membrane. Also some plant cell membranes like plant cell wall, plant cell membrane. So bacteria, this tDNA has to cross the membranes of the bacteria, and also it has to cross the membranes of the plant cell to take entry into the plant cell. Right? So to take entry into the plant cell, this agrobacterium uses the type 4 secretion system. Okay, agrobacterium uses type 4 secretion system. In short, we say it as T4SS. Right? This type 4 secretion system, it is encoded by another kind of Vir gene known as Vir-B. So this is in total encoded by 11 it is formed by 11 proteins, that is from Vir B1 to Vir B11. Okay, it is encoded or it is assembled from 11 proteins. This is assembled from 11 proteins, that is Vir B1 to Vir 11. Okay, and this is a transmembrane complex. So what happens is that the tDNA strand, now we understood that this tDNA strand remain attached with this Vir D2. I label it here, the blue one is D1. So guys, just a moment, I have made a one change. I have just changed the place of Vir D1. It should be afterwards, D1 and D2. D1 and D2. Got it? And this is C1 and C2. Here the C1 and C2 is going to bind. So what we got to know, that when the T strand is transferred, when it is made into single stranded T strand, this D2 remain attached. This D2 remain attached. This is what D2. Okay, and this is tDNA strand. This is where D2 and this is tDNA strand. Okay, so it is made from 11 proteins, and here they are made from Vir B1 to Vir B11. Understood? Now, this T4SS, it is also known as mating pair formation. It is also known as mating pair formation, in short MPF. Another thing is that this membrane spanning complex, it is forming a pore or a channel. Okay, this is forming a pore or a channel through which the tDNA will enter into the host cell. Okay, and this T4SS, it is composed of two components. What are the two components? The one of the component is T pilus, which helps in conjugation process, and another is the membrane spanning complex. Okay, another is membrane associated complex. And it will help in translocation of this tDNA into the host cell, right? This 11 proteins, okay, which make this Vir B, which make this type 4 secretion system, they are encoded by, they are encoded by, they are encoded by Vir B and Vir D4. Vir B operon and Vir D4. So Vir B operon and Vir D4 together, they will make the type 4 secretion system. Understood? And this type 4 secretion system will help in the entry of the tDNA into the plant cell. Now, other than Vir D2, which is entering with the tDNA which is entering with the tDNA into the plant cell, there are other proteins, there are other Vir proteins which will enter into the host cell. Okay, other Vir proteins which will also enter into the host cell. What they are? That is Vir B5, that is Vir E2, Vir E3. E3 is helping the Vir E2 to enter the nucleus. Vir F, Vir F is an F box protein, and they all are translocated. They all of them, they are translocated into the host cell. Okay, and this Vir D2, which we already know now what is its role, this Vir D2 and Vir E. Okay, Vir D2 and Vir E2. Vir D2 and Vir E2, they do not translocate together. Okay, one thing I would like to mention, that this Vir D2 and E2, they do not translocate together. They do not translocate in the bound form. This where D2 is bound to this T strand, this where D2 which is bound to the T strand, they first enter the host cell along with vir E2. Vir E2 and D2 plus T strand will enter separately. They'll enter the host separately and they are going to assemble inside the host cell. That is vir D2. Here is the D2, here the black colored part is the T strand, and this E part, this vir E2, okay, this vir E is going to assemble inside the host cell. This is inside the host cell. Assemble of vir E will be inside the host cell. Got it? Coming to another important Vir protein. So we now understood about Vir E2. So first of all, what is Vir E2? Vir E2 is a single stranded sequence non-specific tDNA binding protein. So Vir E2, it is a single stranded binding protein. It is sequence non-specific. Okay, it binds to tDNA and protects it in the single stranded form. Okay, it will bind to E2 and will protect the tDNA strand. So once this Vir E2 is bound, once D2 is bound, this Vir D2 and E2, Vir D2 and tDNA Vir E2, Vir D2 and Vir E2, they contain the nuclear localization signal. They contain nuclear localization signal, as a result of which this complex, which is Vir D2, T strand and Vir E2, which is known as T complex, this will enter inside the nucleus. This will enter into nucleus. And entry of this into the nucleus, it is again facilitated by another protein, and this is a host protein. Another protein named VIP1, and this VIP1, it interacts with Vir E2. It interacts with Vir E2, is a host protein, and this protein helps into the entry of this Vir tDNA, entry of this genes in this tDNA into the nucleus. So this VIP1 role is that it will help this tDNA strand to take entry into the nucleus. It helps tDNA to enter into nucleus. Got it? Now, once this tDNA strand enters into the plant nucleus, so once this, now in nucleus, what is going to happen? This tDNA strand, once it's inside the nucleus, this tDNA will become double stranded. Single stranded has entered, it will become double stranded tDNA. And this double stranded tDNA, will be then inserted into the genome of the plant. It will be inserted into plant genome. Okay, where it is going to insert? Will be inserted into non-specific region, into non-homologous, it will be inserted into any random position by non-homologous recombination. So it will be inserted into the random position by non-homologous recombination. So if we revise what we studied till now, Okay, if we revise what we studied till now, we studied the agrobacterium mediated transformation in plant cell. At first, there were two genes, excuse me, where A and where G gene, which was constitutively expressed, where A gene has a receptor which will sense the phenolic compounds released by the wounded plants, that is acetylserinone released by the wounded plants, where A and where G acts in the two component signaling system, where A is a sensor kinase, where G is the response regulator. Once where A sends the acetylserinone, it will get autophosphorylated on its histidine residue, and then it will transfer its phosphate group to the aspartate residue of Vir G gene. Okay, this Vir G gene, once it is activated, it acts as a transcription factor. Its product will act as a transcription factor, which goes and binds to the promoter of other Vir operons and in turn activate other Vir genes. Okay, well, how it is going to activate other Vir genes? By binding to the promoter of other Vir operon, right? Now, first of all, in the first case, now I am going to mark these steps. Number one is that where D1 and D2 genes are activated, they are going to generate the single stranded tDNA. They are going to generate single stranded tDNA. It is known as T strand. This where D2 is an endonuclease. This where D2 will remain attached to the nicked tDNA via the tyrosine residue, which is known as phosphotyrosine bond. Right now, this what is the role of Vir D2? Why it remain binds to the T strand? Because it was proved that this might be protecting the tDNA from exonucleolytic attack. Okay, from exonucleolytic attack. Also, it remain binds to the 5' end, which says that this is the leading strand from where the transfer will take place. Okay, this is the side from where the transfer will take place. Next comes the role of Vir C1 and C2. This C1 and C2 binds to the cis element near the right border, and this cis element is known as overdrive sequence. Okay, this overdrive sequence, what its role? That it helps in efficient tDNA transfer. Next we come to Vir B and Vir D4. Vir B and D4 role is to form what? Is to encode Vir B proteins, that is from Vir B1 to Vir B11. 11 proteins together will make the type 4 secretion system we say as T4SS. It is going to form the pore or channel through which the tDNA will transfer into the host cell. Okay, this is also known as mating pair formation, MPF. T4SS has a T pilus and membrane associated complex. Two components are there. Apart from that, there are several other Vir proteins which are translocated into the host. This is D5, E2, E3 and F. E3 is a helper of E2. I write here E3, helper of E2. And what is E2? E2 is a single stranded binding protein. E2 is a single stranded binding protein. Next we come to with D2, E2 done. With D2 and E2, they will enter into the host cell separately. Okay, and they get assembled. This is the host cell. This is the host cell. They will assemble where D2 and E2 assemble in host cell. Enter separately, assemble in host cell. E2 is said to protect the tDNA from the digestion by the nucleases, and this D2 and E2, they contain the nuclear localization signal which allows the tDNA strand to take entry into the nucleus. This entry is facilitated by another host protein named VIP1. This VIP1 binds to which protein? It binds to E2 protein. okay, and helps the tDNA strand to enter into the nucleus. Inside the nucleus, this single stranded tDNA will become into double stranded tDNA. It is then inserted into the plant genome into random position by non-homologous recombination. Okay, I hope this part of agrobacterium transformation is clear to you. If you have any question, your comments will be uh, appreciated in the comment box. So please give your feedback. Any questions are welcome. Thank you so much. Please like and share the video, and please subscribe to my channel. Thank you so much.